this is for the meiosis narrative simulation, and I'm gonna start. So then we begin an interface for meiosis, the start of meiosis, and the cell grows in the G1 phase and also matures in the G1 phase, and then the chromosomes replicate to form sister chromatids, and this is in the S phase for sister chromatids. And then the formation of S is all X here, and then it will duplicate in G2, and then for meiosis, and this is meiosis 1. So meiosis 1 begins in prophase 1, which is the start, and then and then chromatins condense to form chromosomes, in which they become visible. Let me put that in this is a nucleus for it. But then the nucleus disappears, and then centrals appear as well. Let me organize these. The centrals appear. These paper slips are centrals, and something called synapsis occurs, in which a homologous chromosome begins to pair up, and then they form like a tetrads, which are these things. And then crossing over will eventually occur, and then the homologous chromosomes, these, exchange segments. So like, example, this goes here, blue and red goes here, and then I'll just get rid of purple. And that is the end of prophase 1. And then next is metaphase 1, in which the centrals move to the opposite ends. So centrals will move here, and spindle fibers from the centrals attach to the, like that, like that. And my fingers are like this spindle fibers. And they attach to the centrum of the chromosome. And then the homologous chromosome pairs up like randomly in the middle. So yeah, like that. And which is called independent assortment. And then spindle fibers begin to shorten and separate the chromosomes apart, and then they separate to opposite sides. So then this blue and red goes over here, and then the green and yellow go over the opposite ends with the centrals. That is the end for anaphase, and then it begins the telophase 1, which is when the nucleus reappears. This is the nucleus, and then this is the other nucleus. And then I'll keep it there. And then the cell membrane of the cell pinches forward like that, and then inwards as well. And then cytokinesis 1 completely separates the cell, resulting in two cells. So that and that are 2 cells. And then for meiosis 1, we'll do it like this. For meiosis 2, and then the second process. This would be the new cell that we have. And then let me bring these in. Okay. So then the nucleus disappears. So disappears. And it begins or goes on to metaphase 2, in which the centrals right here move to opposite ends once again, and the spindle fibers, as mentioned before, from the centrals attach to the centromere of the chromosomes, and then those chromosomes begin to line up in the middle, like this. Oh wait, this is a different colors. Here. Different. In the middle. And then that goes on to anaphases 2, in which the spindle fibers begin to shorten and separate the chromosomes to opposite sides towards the centrals. And then this will go here, 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 and like here, like that. And then this moves on to telophase 2, in which the nucleuses reappear. So here and then right here. And then, as said before, the chromosomes like kind of decondense again, and then they come back to chromatins, and the cell membrane bridges forward. So it can begin cytokinesis 2, in which it completes separation, and then we have 4 like different haploid cells that are genetically different as well be created. That is it for meiosis narrative.